this broader impacts project, I realized that one of the things that I wanted to do was to introduce some of the concepts and the data that we used and then collected in Mongolia this summer at the high school where I used to teach in Philadelphia. And we will talk a little bit more about that. And so I designed a Food Webs mini course this fall and taught it at that school. And these are the students in the school, and they will introduce themselves shortly. And we're going to talk today in sort of two directions. It's a multiple personality presentation. We're going to talk a little bit about the education component and also a little bit about the research component. And you may find it confusing, which one do you pay attention to, which one do you ask questions about, and you can pay attention or ask questions about whichever section you want, and at the end you can ask questions of them or of me or both, however you want. So just so you know, everything is very easy. And so, Mike, please stop.
Hello everyone, I'm Aisha Clark, and I'm a senior at Mason Jordan College campus. Today we're going to be presenting to you Exploring the Depths of Mongolia. We had a brief over our course for the first semester of school. It was main ecology course, so we're going to present to you the information we obtained over the course of this session. And we're going to introduce ourselves as the Mason Jordan College campus Advanced Ecology Group.
My name's Aldo C. Liam, and I'm a freshman.
My name is Nick Marcico, and I'm a senior also.
My name is Annie Nguyen, and I'm a senior.
Jalila Lewis, and I'm a senior.
Kelly Nguyen, I'm a senior also.
I'm Ahmed, I'm a senior.
My name is Miranda Berkman, I'm a senior.
My name's Anna Nguyen, and I'm a freshman.
My name is Hannah Hunt, I'm also a freshman.
And
I'm Alfie Amilko, and I'm a freshman.
So after schools on Tuesdays this past semester, us 5 freshmen and 7 seniors have gathered together to learn about food webs and isotopes instructed by Ms. Young.
The students were chosen based upon an application they had to fill out. And the activities we did over the course were we designed graphs using a program called R, and we learned about isotopes and food webs through Ms. Young's lectures. And we took a field trip to a Rutgers Marine Steel Station, and we just dissected like seagulls.
And the program originally had was originally conceived as a pen pal program, and so I set it up so that every student here had a pen pal in Mongolia. But of course, because of both Mongolian time frames and Mongolian mail rates, everybody here has sent their first letter, but we have not yet gotten any letters back. So hopefully that will continue this way. But that was sort of part of the frame. But sort of separate from the pen pal component, what I had envisioned was being able to introduce some concepts and tools used in food webs. So got content analysis, stable isotope analysis, and then with that introduction, then do some preliminary analysis of the data that I collected in Mongolia this summer with that background. And so I want to give a little bit of background on Mongolia so that you have that as part of it. So Mongolia is a landlocked country. It is squished there between Russia on the north and China in the south. It's the most sparsely populated nation in the world. There are about 2.75 million people there in an area just smaller than Alaska. It's primarily a steppe landscape, and so the primary occupations are related to herding livestock. And Genghis Khan, if anybody was here for Julia's talk last spring, Genghis Khan is a hugely important cultural and historical figure. So there are statues of him everywhere, and many companies and brands are named after Genghis Khan. Mongolian is its own ethnic group, and it's also its own language. And Mongolian is the most well-known language in its language group. It is not related to any other Asian or Central Asian languages that any of us have ever heard of. And it's written now in Cyrillic after many years of communist occupation, although originally there was a more traditional vertical script. And the capital, Ulaanbaatar, which you can see just in the center of the map on the bottom, is home to a few over 1 million people, and so a little bit less than half the population. But one thing that I observed in Mongolia, and that Olaf has also commented on in terms of his experience, is that unlike other rapidly urbanizing regions, the people who live in the city in Mongolia maintain a really strong relationship with the countryside. So they go back frequently, they send their children every summer. And so the relationship between the city and the country seems stronger there than it has been in other places that I've been. So I was there this summer for 5 weeks. Olaf has a 3-year grant, so this last summer was the first summer of 3 at least that we will be spending there. And we worked in a team of about 15 people. It was about half Mongolian and half people who lived in the U.S. Not all of the people who lived in the U.S. were American. But, and And it was a mix of principal investigators, graduate and undergraduate students, technicians, fishing guides, and broader support staff, which includes drivers and cooks, although it's not just driving and it's not just cooking in Mongolia. And I would say that actually that group of people, most of them Olaf had relationships with previously because he did a chapter of his dissertation there. And the fact that we have strong relationships with our Mongolian colleagues, I think, contributed very, very strongly to the success and ongoing nature of this project. And I think the people working there, we get along with and have good personal and professional relationships with, and I think that really makes this project float, and that is part of why Olaf is really committed to continuing to go back. So we did most of our work on Lake Hufsagul, which is a lake tucked up there in just south of the Russian border, and it is a lake that is longer than it is wide. And if we go to the next one, it is the 17th largest lake in the world by volume. It's fairly deep, fairly old, and fairly high altitude. It is an oligotrophic lake, meaning it's very, very low nutrient, so it's very clear. So there's not a lot of planktonic life going on. There's some, but not a lot. We measured a Secchi depth this summer of 16 meters, which is really, really deep. What a Secchi distance would be able to see it. It's also dimictic, meaning that the water mixes vertically twice a year, in the fall and the spring, and I'll talk more about that in a moment. It's ecologically interesting because there are only 9 known species of fish in the lake, and there are no known introduced species. It's very isolated. To get there, it was 3 days in a van on unpaved roads. And it is designated a national park and an international long-term ecological research site. And as a result, there's minimal development. It's not no development, but there are some resorts. So that picture on the bottom, you can kind of see there are some houses and some yurts, which is the Mongolian word for yurt, where Mongolians, really only Mongolians, go to vacation there. And you can see there's some grazing around parts of the lake. And we saw evidence of logging within the national park. So the national park regulations are clearly not strictly enforced because you can log in the lake. And there's no commercial fishery. We did see some poached fish. So those fish in that picture are ones that we saw when we were driving along the road that people had caught illegally and were planning to sell. But there's no large-scale commercial fishery. So this lake is of interest to study for a number of reasons. Due to its isolation and the small fish species complement, it offers a unique place to ask fundamental questions about aquatic ecology. And because we think that with such a simple system, we might actually be able to characterize the whole thing, which is not true, say, in Lake Tahoe or Lake Superior, similar lakes that are well studied in the U.S. that have many, many epidemic impacts, lots of development, lots of invasive species, many other factors at play. And so one of the questions that Olaf has raised is an interesting one about the lake, is the relationship between ecosystem size and food chain length. So David Post's famous paper about where he looked at many, many lakes in New York State, and he measured both their volume and the maximum trophic level in those lakes, and he found that there was a positive relationship, that as the lakes got bigger, the maximum trophic level increased, the food chain length increased. And so his data are in diamonds on that graph, but that black dot over there is Lake Hufsagul. So these are some interesting questions. Why is it there? Is it because Lake Hufsagul is very, very, very unique in the world? Does it have to do with Lake Hufsagul's otherwise isolated nature, its lack of epidemic effects? So this is one reason why this lake is interesting to study. It's also an interesting, region is an interesting place to look at climate change. Because of its isolation, there are not a lot of other impacts. So changes we might see in this area could actually be due to climate change rather than being due to other factors. So I want to talk a little bit about some possible hypotheses about climate change and its relationship to freshwater systems. Yeah. So, as the oceanographers here know, the relation to the seasonal and vertical layering of water in aquatic systems is a really important part of their ecology. And climate change it's hypothesized that climate change may affect some of that layering in multiple ways. So just as an introduction to thermal layering in lakes. If we start with the summer, you can see in the diagram, in the summer the water on top is warm, the water on the bottom is cold. So those layers generally stay distinct. The hot water stays on top, the cold water stays on bottom. That's thermal stratification. In the fall, the water on top cools down, and so because the water temperature becomes more unified throughout the body of the lake, the water mixes vertically. So there's mixing going on. And then in the winter, the lake freezes over, the top is very, very cold, the bottom is a little bit less cold, and again, the layers of water stratify out. In the spring, the top of the water heats up again, the lake temperature unifies again, and the water mixes vertically again. And that spring mixing is a critical feature of aquatic ecology. That spring mixing brings the nutrients up from the bottom to the top part of the lake and provides the nutrients for the spring phytoplankton bloom. The phytoplankton bloom in the spring, the zooplankton eat those blooming phytoplankton, the zooplankton explode, and then all the other organisms eat the zooplankton. So that spring mixing is really key, and there are questions about if temperature increases, will the spring mixing happen earlier? Will the earlier spring mixing cause an earlier phytoplankton bloom? Does that mean that the zooplankton will reproduce at the correct time to correspond with the phytoplankton bloom? That is the match-mismatch hypothesis. Some other questions about stratification and increased temperature. Does it mean that the water gradient, the temperature gradient in the vertical temperature gradient in the lake will be more extreme, so the stratification will be stronger? Does that mean that there will be thermal zones in the lake where fish do or do not go? So fish who prefer cold water will not go into the warmer sections. Will that affect their interactions or their predation or their prey interactions with other fish because of the thermal stratification? There are questions also about whether if we increase the permafrost from thaw, and some of the researchers at Penn who are working in Lake Lucille have shown that the permafrost is indeed thawing. Whether that will increase erosion, that will increase runoff, whether that will increase the cloudiness and turbidity in the lake, if that will change the photosynthesis that goes on in the lake. So these are some of the things that people are talking about in terms of how will climate change affect freshwater systems. So with all of that as context, the project this summer was much less ambitious, was much smaller. And it was simply to create a baseline and descriptive food web for the lake using C13 and N15 isotopes. And the hope is that we can both use this to characterize and describe the lake itself, and perhaps this may also be able to be a baseline for looking at climate change in the region in the future. So we were using C13 and N15 isotopes because they can tell us different things about food webs.
So delta C13 is an isotope that determines the carbon source of an organism. In other words, it tells us where the food comes from. So this is a graphical representation of how delta C13 shows us the sources of the food. So in the x-axis are the values of delta C13, or in other words, the ratios. In the y-axis are various sources. The biggest takeaways of this graph is freshwater phytoplankton, which is in the range of -24 and -27, and that indicates that it's very benthic. And in case you're not familiar with the term benthic, pelagic. Never mind. Pelagic. So pelagic organisms feeds off in the opening of the lake, so in the middle of the lake. And in contrast to pelagic, the benthic algae, which is in the range of -14 and -17, they're very benthic because they feed off at the shore of the lake. So in other words, they feed off where the water is very shallow.
And delta N15 is basically the organism's trophic level. So meaning the higher the nitrogen ratio is, the higher they are on the food chain level.
So this graph shows the delta N15 and delta C13 values of two types of birds, the cormorants and jacanas. And from the delta N15 values, you can tell that the cormorants they feed on organisms from the higher trophic levels, followed by the second group of jacanas, then the first group of jacanas. And from the delta C13 values, you can see that the range of sources of carbon for the cormorants is very wide, as compared to the range of the sources for the jacanas, which is narrower, which means that they have a much more varied diet than the jacanas.
So this summer, in order to collect our C13 and N15 samples, we sampled at 10 different locations around the lake. And we sampled fish, invertebrates, and primary producer samples at those locations. And we use a variety of different collection methods to do that sampling. We use gill nets and horizontal and vertical gill nets, and we did some staining. We used hook and line and minnow traps. We did some server sampling, and we did do some tow nets for plankton, tow nets for plankton, and we did a lot of hand collecting, picking stuff up. And then when we had collected those, we dried them in a solar oven. And the solar oven ranged from 50 to 90 degrees Celsius, depending on how sunny it was. And then we sent them when we came back to the UC Davis Naval Isotope Facility for analysis. And we're lucky to have data from 2005 and 2009, and I'm really excited to look at that, but we're not going to focus on that today. We're just going to do a little bit of preliminary presentation of some of the 2011 data from this summer.
So back to the Delta N15, if you can see, there's a big gap between the invertebrates and the fish, and that makes the fish are more up top and the invertebrates are more down the bottom. And that makes sense because the fish are on a higher feed level, so they feed on more organisms that are on their same trophic level, and the invertebrates feed on organisms that are on their lower trophic level.
And as you can see that the snails and the zooplankton, they feed on lower levels, so, and zooplankton are more pelagic, so that means they feed out in the open ocean. And so snails are more benthic, so they feed on shallow water. But as you see for the fish, they're not very spread out. Like graylings and minnows and they feed on more benthic level, but also for the grayling you can feed on more pelagic also. And for the birch, it's just a lot more pelagic than all the other fish.
So we've only just barely scratched the surface on this data. I think there are a lot of directions we can go with it. One of the things that Anna and I actually were talking about last week is that some of the N15 levels or ratios were a lot higher than I was expecting, and we talked about how that might be influenced by some of the land use. Maybe the perch may, I have to go back and look, but may have been collected in places where there was a lot more grazing, and whether the grazing and livestock runoff may be elevating those N15 levels. Two of the other students there last summer did linnet and grayling, the two kinds of fish, did diet analyses, and I'm interested, their data are coming. I'm interested in looking at their data in relation to the stable isotope data. I think next would be to learn about stable isotope mixing models, about which I know nothing, but that's kind of the next project. And then to start thinking about climate change hypotheses. So that's sort of some next steps on the research front.
So furthermore, we would like to create data posters about our Black sea bass dissections, and we would also like to enroll in a spring course in which we do more hands-on analysis and activities.
Now that this course has come to an end, we have learned many things, as you can see thus far. Although there needs to be improvements, there were the many positive aspects that we've come across: meeting new people, learning more information, and performing the dissection. Overall, everyone has agreed that this was a great experience. I would recommend it to anyone.
From an instruction point of view, I had a lot of things that I learned from this. I mean, the first one was that it ended up being a lot more challenging than I expected. I knew it would be hard. It was harder than I thought to teach meaningful content about a place that was really far away where the students had never been, and about data that the students didn't collect. So I think that was one of the challenges. I had forgotten, this happened to me when I taught AP as well, but I'd forgotten, so I recruited sort of the best and the brightest, and it has been totally awesome. But what it meant is that I forgot that I needed to really scaffold the teaching anyway, because the content is really hard. So like doing stable isotopes with people who have never taken chemistry that's something, right? So, and then I sort of forgot that I needed to do more scaffolding than I did. And I also didn't realize, so we only met once a week, and that actually meeting once a week is not enough time to do really hard content. And so that was interesting to me, and I don't quite know how to reconcile that part of it, but that was good to know. One of the things that happened was that the students introduced me to Prezi, which is this software that we use to do the presentation. And it was great to learn a new software, but it was also, Prezi was really great for group work. So all 10 of us could be simultaneously on this at once, and we could all edit it at home, and then we'd come on, and new things would have happened, and new things would have changed. So that was a really great tool for group presentations that I hadn't known about. And then the other thing is that I feel like there is some pressure in graduate life to work nonstop on your research, and I do feel strongly that doing this kind of project increases my productivity at other times than it would otherwise, and I feel like that's sort of worth noting in terms of planning my time in the future. We have many people to thank. There were was a large research crew in Mongolia that helped us, and many people at Rutgers, both sort of on this campus and then at the field station for our trip to the field station. I'm funded this year by the field station and INC, SGA, and NSF, and we also have a bunch of people thank at school.
We would like to thank the following people at Mastery for allowing this course to happen. Christy Fruit, our assistant principal, Terry Hines, the director of operations, Nikki Foy and Ryan Kushulas, our teachers, and Matt Troha, our principal.
And Matt, I mean, among other things, drove everybody here, so Pastor Vance, that was a really big thing that we not have been able to happen otherwise, so yes, thank you. So now we'll take questions.